Hey, welcome to another episode of Following Science, the podcast where I dive deep into the latest breakthroughs and insights from the world of science. I'm your host, Rutu Parl, and today we're going to be exploring the genetic underpinnings of Alzheimer's disease, particularly focusing on a recent study that sheds new light on a specific genetic variant. Alzheimer's disease, often simply referred to as Alzheimer's, is the most common form of dementia, affecting millions of people worldwide. It is a progressive neurological disorder that leads to memory loss, cognitive decline, and ultimately the inability to carry out simple tasks. While much of our understanding of Alzheimer's comes from studying its symptoms and progression, recent research has provided a clearer picture of the genetic factors that contribute to the disease. Now, to understand Alzheimer's disease, it is essential to distinguish between its two primary forms, early onset and late onset. Early onset Alzheimer's, though less common, can appear in individuals as young as their 30s or 40s. This form of disease is often genetically determined and can be linked to specific genetic mutations, such as those in APP or PSEN1 and, of course, PSEN2 genes. These mutations follow an autosomal dominant inheritance pattern, meaning that they just have one copy of the mutated gene that can lead to the development of the disease. Another group at high risk includes individuals with Down syndrome, who almost invariably develop Alzheimer's as they age due to an extra copy of chromosome 21, which carries the APP gene. Now, most Alzheimer's cases, however, are late onset, typically developing after the age of 65. Unlike early onset forms, late onset Alzheimer's is not directly caused by a single gene mutation, but rather involves a complex interplay of genetic, environmental, and lifestyle factors. Now, one of the most significant genetic risk factors for late onset Alzheimer's is a variant of the APOE gene, known as APOE4. The APOE gene exists in several forms, or alleles, with APOE3 being the most common and neutral regarding Alzheimer's risk. APOE2, on the other hand, appears to provide some protective benefits against the disease. But it's the APOE4 variant that has garnered the most attention. Now, individuals who inherit one copy of APOE4 have an increased risk of developing Alzheimer's, and those with two copies, referred to as APOE4 homozygotes, are at even higher risk. In fact, APOE4 homozygotes, who make up about 12% of the population, are estimated to have a 60% chance of developing Alzheimer's by age 85. This significant risk has prompted extensive research into understanding how the presence of APOE4 influences the onset and, of course, progression of the disease. Now, a recent study led by Dr. Fortea and Victor Montal at the Sant Pau Research Institute in Barcelona has provided groundbreaking insights into the role of APOE4 in Alzheimer's disease. This study, published in the Nature Medicine on May 6 of 2024, analyzed post-mortem brain pathology from over 3,200 individuals of European descent, focusing on those with different versions of the APOE gene. The researchers also examined clinical, pathological, and biomarker data from more than 10,000 people across 5 clinical studies. The findings were striking. Nearly all APOE4 homozygotes in the post-mortem database exhibited Alzheimer's brain pathology from age 55 onwards. This was a stark contrast to about half of those without the APOE4 variant. Furthermore, APOE4 homozygotes consistently had high levels of Alzheimer's biomarkers, such as amyloid beta, starting at age 55. By age 65, almost all had abnormal amyloid beta levels in their cerebrospinal fluid, and 3/4 had detectable amyloid plaques in brain imaging. Now, these biomarkers are crucial because they signal the presence of Alzheimer's-related changes in the brain long before clinical symptoms appear. For APOE4 homozygotes, the age of symptom onset averaged around 65, with mild cognitive impairment typically diagnosed by age 72, dementia by age 74, and death occurring around age 77. These timelines are about 7 to 10 years earlier than for individuals without the APOE4 variant. The study also found that variability in the age of symptom onset was less pronounced in APOE4 homozygotes, making the progression of the disease more predictable compared to those without the variant. This predictability is similar to what is observed in other genetically determined forms of Alzheimer's, such as autosomal dominant Alzheimer's disease and Alzheimer's disease and Down syndrome, or DSAD. Now, findings of a study researching this gene specifically represents a significant shift in our understanding of Alzheimer's disease, particularly in individuals with 2 copies of the APOE4 gene. The research suggests that being homozygous for APOE4 could, in fact, represent a distinct genetic form of Alzheimer's. This means having 2 copies of APOE4 is not just a risk factor, but a causing factor for Alzheimer's in the studied population. Now, the implications of this are profound, as it could change how we approach diagnosis, treatment, and research for Alzheimer's disease. The study indicates that future research might need to focus on developing specific diagnostic and therapeutic techniques for those who have only 2 copies of the APOE4 gene. However, it's important to note that much of this data in this study comes from individuals of European descent. Further research is of course needed to determine if these findings hold true across different ethnicities. Now, another significant aspect of this research is the potential for personalized medicine. Understanding that APOE4 homozygotes follow a more predictable disease course opens the door for targeted interventions and treatments. For instance, NIH is currently funding studies to explore potential treatments specifically for individuals with 2 copies of the APOE4 gene. Additionally, increasing diversity in Alzheimer's research is crucial to ensuring that findings are applicable to all populations, not just of European descent. This study also raises important questions about the ethical considerations of genetic testing for Alzheimer's risk. While identifying individuals with a high genetic predisposition to Alzheimer's could lead to earlier and more personalized interventions, it also brings challenges related to genetic privacy and psychological impacts. Now, as we continue to unravel the complexities of Alzheimer's disease, studies like this one by Dr. Fortea and Montal provide invaluable insights that bring us closer to understanding and ultimately combating this devastating condition. By recognizing the distinct genetic profiles of individuals at risk, we can move towards more effective and personalized approaches to treatment and prevention. Now, as I wrap up this episode, I just want to thank you all for joining me on this episode of Following Science. If you enjoyed this episode, please subscribe, leave a review, and share it with your friends and colleagues. Stay curious, stay informed, and keep following science. Until next time, I'm your host, Rutu Raparo, signing off.